What happens when you follow the rabbit hole of the unknown? Brace for impact because you never know what you will discover on the other side. Why do people love animals so much? Well, it's similar to how humans are biologically connected to loving their children. Animals need our help, appreciate our help, and in turn, they provide us with their expression of affection. As people, we also have less expectations from animals than we do from other humans, so we tend to be more satisfied with their presence. In 1996, Dolly the sheep was not the first cloned mammal, but what made Dolly so special is that she was made from an adult cell, which no one thought was possible at the time. Dolly lived for 6.5 years and gave birth to 6 lambs. There was a theory that Dolly's age had accelerated because she was cloned with adult DNA. However, Dolly later contracted a virus at the Roslin Institute where she lived and developed lung cancer. She was eventually euthanized and now on display at the National Museum of Scotland. A pair of cloned calves were born in Japan in 1998, 2 years after Dolly. They were named Kaga and Nodo. Both died of old age at the ages of 20 and 21. The average lifespan of a cow is 20 years old. Since then, a total of 14 cloned cows have been produced, but research was cut back after the distribution of cloned cow meat was restricted in 2009, but the study of the lifespan of cloned cows has continued. The first cloned dog was born in 2005, an Afghan hound named Snuppy in Seoul, Korea. Snuppy was cloned by using a cell from an ear of an adult Afghan hound, using 123 surrogate mothers, and only 2 produced pups, and Snuppy was the sole survivor. Seoul National University was awarded Time Magazine's Most Amazing Invention. Today in South Korea, Sohum Biotech Research Foundation will clone dogs for customers for approximately $100,000. The most common breeds are Labrador Retrievers, Yorkshire Terriers, and Jack Russell Terriers. Sohum believes they are healing broken hearts by bringing back your beloved pet. After your dog passes away, wrap the entire body with a wet towel. Place in the fridge, not the freezer, to keep cool. Then follow the biopsy instructions on the company website within 5 days to successfully extract live cells. Here is an example of how to extract DNA from cells. Step 1, spit into a cup 3 to 4 times. Step 2, add a dash of salt to the saliva. Salt swells up the cells, then stir. Step 3, add 1/4 teaspoon of dish soap to burst open the cells, then stir. Step 4, cells from the spit are releasing DNA. Step 5, squeeze 1/2 teaspoon of fresh pineapple, then stir. Bromelain is in pineapple and a type of enzyme known as protease. Bromelain breaks down proteins and releases the DNA with minimal breakage. Step 6, add 1 tablespoon of alcohol, then stir. Alcohol will precipitate the DNA and make the strands clump together so they are visible to the naked eye. Step 7, thin strands of DNA are produced. So why is there a deep desire to clone animals? One reason could be that 99% of the 4 billion species that have lived on Earth have now gone extinct, and new species have formed. If this study is true, this would suggest that by the end of the year 2100, almost half of the planet's species will disappear. And some believe creating one of the greatest ecological destructions known to mankind. Some scientists believe that resurrecting extinct species is impossible. There was a study done on the extinct Christmas island rat that proved bringing back the woolly mammoth and other extinct species is not possible without very important differences. Therefore, it is impossible to bring back extinct animals exactly as they once were. In the case of the Christmas island rat, researchers were able to recover a high-quality genome from preserved specimens, but it was impossible to recreate several key genes needed, making the rat different in crucial ways. The Christmas island rat would be missing important characteristics from its extinct form. Thomas Gilbert with the University of Copenhagen in Denmark says, the issue is that old DNA can break into many tiny pieces that can be impossible to duplicate or resemble. For example, the Christmas island rat went extinct in the early 20th century, and researchers were able to resemble a majority of the DNA by using the Norway brown rat as a guide, but it could not be assembled 100% the same. Gilbert said every bit of DNA they could recover, they got, but there's a 5% fraction they could not make sense of. Meaning the crucial parts of the extinct genome that are different from most living relatives are the most difficult to match and resemble. That missing 5% are genes that have rapidly evolved and therefore what make the closely related relatives different from extinct species. It's similar to a missing piece of a puzzle. Gilbert said researchers were able to recreate 50% of the genes for the Christmas island rat, such as the hair and ears, so it looked similar to the species, but several other genes like the immune system, sense of smell, were partially matched. Smell is actually an important gene because it helps play a vital role in determining behavior like finding food, avoiding predators, and selecting mates. The outcome would be very different from the original Christmas island rat. Beth Shapiro at the University of California, Santa Cruz says the study of the Christmas island rat proves the distance between extinct species and living relatives and the genomes that can't be correctly assembled, but still possible to create species with physical and behavioral traits that reflect that of the extinct species. Shapiro went on to say that this research shows that reconstructing a genome using gene editing cannot be 100% identical to extinct organisms. In regards to the woolly mammoth, Thomas Gilbert said, if you think you are going to create a mammoth that's exactly like the mammoth that went extinct, well, you're not really going to be doing that. Gilbert went on to say that he isn't against de-extinction and agrees it is feasible to create animals that can perform the roles extinct animals had in our ecosystem, but the end product will be different. Researchers have been trying to resurrect extinct animals by sequencing the DNA in preserved samples, by genetically editing the genome and using a closely living relative and making it like the extinct species. USA-based company Colossal currently has a team working to bring back the woolly mammoth and has raised millions of dollars to fund the project. The University of Melbourne in Australia is currently working toward resurrecting the thylacine, also known as the Tasmanian wolf or Tasmanian tiger. Scientists believe bringing back the woolly mammoth could help the Arctic tundra environment and help stop the release of greenhouse gases from the ground and reduce the future emissions and temperatures from rising due to climate change. Reestablishing this ecosystem could have a cooling effect on the climate. Recreating a grazing ecosystem will trample down the snow during the winter and deeper freezing of the permafrost during the winter. Protecting the Arctic's permafrost, the world's largest carbon reservoir, releasing CO2 and methane into the atmosphere. In terms of cloning the woolly mammoth, the project would consist of finding a closely related relative and making a new version of the Asian elephant that is cold resistant and capable of thriving in an ecosystem and comfortable in minus 40 degrees weather at months at a time. In regards to the thylacine, also known as the Tasmanian tiger, the most closely living relative was thought to be the spotted quoll, but researchers now believe it is the fat-tailed dunnart, which is the size of a mouse. In Tasmania, Australia, humans killed the thylacine and drove the species to extinction, with the last living Tasmanian tiger dying in 1936. Now there is an overabundance of disease-infested marsupials because there are no predators keeping the marsupial population in check, and the environment is unbalanced. Bringing back the thylacine could help balance the massive problem with overpopulation in Tasmania. Plus, researchers want to give the thylacine a second chance at life. What are your thoughts on genetic engineering, and do you believe the risks could help save our planet? Thanks for watching, and stay safe and well.